Okay, so welcome to the second of two videos on DNA
It's
and
9 o'clock.
oh, thank you, Jesus. Um, to the start that again. The second of two videos on DNA and biotechnology. So we finished last time with the slide that you see in front of you right now on the genetic code. So we'll move on from here. The one thing I wanted to add, though, was that notice that AUG is the start codon. So that's the first one in every mRNA strand. And then any of the three that you see up here are what we call stop codons. All right, and those are found at the end of the mRNA molecule. Okay, so remember that the two major portions of gene expression are transcription and translation. So let's look now into transcription in a little bit more detail. All right. Transcription is essentially taking a DNA molecule and using it as a template to make a messenger RNA molecule. All right. So the first thing that has to happen is that you kind of unzip the double helix of the DNA molecule a little bit somewhere in the middle, all right where a gene. So, for example, I know this isn't 3,000 base pairs, but let's pretend from here to here represents one gene. All right. It's in reality much longer than that, but let's take it from there. All right. Um, what's going on in this scenario is that transcription is taking place. All right, the messenger RNA nucleotides are being joined together by an enzyme called messenger RNA polymerase. Um, so that the sequence that they have is complementary to the DNA. All right, and this brown egg thing is representative of that enzyme RNA polymerase. The green, of course, represents the DNA template strand, and the pinkish purple here on this side represents the messenger RNA transcript. All right, that's what we call it, the transcript. All right, and once the mRNA strand is complete, it is ready for processing, and then it will move out into the cytoplasm. All right, the messenger RNA is processed before it leaves the nucleus. And then once it's processed, it leaves the nucleus and goes out to the ribosomes to be translated, which we'll get to in a minute. Remember, again, that every three bases on the mRNA molecule is called a codon. And each codon, based on the previous slide, codes for a very particular amino acid. Okay, so let's talk about what happens during the processing. All right, this takes place, remember, during or inside the nucleus. So right after transcription, but before translation. And the DNA molecule is represented here on this line, and the mRNA molecule that is produced from it is represented here. Now, notice that there are a couple of things here. Each of these little E's represents what we call an exon, E-X-O-N. All right, and each of these little I's is called an intron. And essentially, processing does three things to the newly made primary RNA molecule. The first thing it does is the one end of the RNA molecule is capped, and the cap is represented by this yellow thing that almost looks like a pencil eraser in this diagram. The second thing is that all of the introns are removed. They're cut out. They're excised. So finally, all that's left is exons, and you can see that here. And then last but not least, you add a polyadenine tail. All right, so in other words, a long tail of lots and lots and lots of adenine nucleotides. All right, so once those three things have been completed, the processing of the mRNA is also completed, and now the mRNA molecule is ready to move out into the cytoplasm. Now, just as an interesting aside, it has been discovered in certain genes that if you cut out one combination of introns, you will produce a certain protein. But if you cut out a different combination of introns, you can produce a different protein. And so essentially, one gene is potentially responsible for producing multiple proteins. All right, that's kind of an interesting discovery that has been actually recently made. Okay, so the next step, the second main step in this process, is translation. And again, we can break this down into smaller steps. So in translation, the first thing that has to happen is what we call initiation. And in this case, the messenger RNA molecule binds to the small ribosomal subunit. All right, each ribosome is made of two proteins, a smaller one and a larger one. And when the messenger RNA binds to the smaller one, the larger one then associates with it, and they come together to produce a complete ribosome. The second step is what we call elongation. And in this case, this is what's going on here is we're simply adding the amino acids to make a longer and longer and longer polypeptide. The way that this happens is that somewhere out in the cytoplasm, the transfer RNA molecule picks up an amino acid. And remember that the transfer RNA has an anticodon that is complementary to the codon on the messenger RNA. So once they match together, the transfer RNA anticodon binds to the mRNA codon, and the amino acids line up, and then they're bound by dehydration synthesis. All right, and they form a polypeptide once it gets long enough anyway. Now, at some point, however, there is a stop codon on the mRNA. And what that does is it causes the ribosome to disassociate and fall off of the messenger RNA molecule. All right, remember, if we go back a couple slides, all right, these three combinations, whoops, too far. These three combinations represent the stop codons. So once the mRNA is processed until it reaches UAA, UAG, or UGA, that will end the translation process. Okay. So what does that actually look like? Well, first, notice that we have two small proteins. This is the small ribosomal subunit, and this represents the large ribosomal subunit. All right, and these are, again, two different proteins. But once we have a messenger RNA that finds a small ribosomal subunit, we undergo initiation. And so the larger subunit then joins or associates with the smaller one. All right, and again, we call that initiation. So they all come together. Now, in the second phase, elongation occurs. And in the first stage, the tRNA polypeptide is at the P site. Now, the P site you can see here, right, is the one on the left in this diagram. So this is the P side, and this is the A side, all right, from this diagram over here. All right. So in the first stage of elongation, a tRNA polypeptide is at the P site. All right, and you can see the polypeptide. Notice that we have methionine, which is the start codon and the first amino acid in every one of these polypeptides. In any event, notice that the transfer RNA in the P site is still connected to the polypeptides. What happens is that now the valine, and I can't quite read that one, ASP maybe? Aspartame? I'm not sure. But anyway, this one, the bluish green one, will associate with the reddish one over here. And notice that they are bound in this picture. So what happens is the whole thing shifts over. And so the transfer RNA that was in the A site moves to the P site once the amino acids have bound together. And the one that was in the P site is then ejected from the ribosome. All right, and that's the second stage of elongation. So the ribosome is moved to the right, and the tRNA polypeptide at the P site is now no longer by one amino acid. But one of the tRNAs is now outgoing. And notice, though, that another one with an anticodon to match the codon is coming into the A site. Now, at some point, you will have a stop codon move through the ribosome. And once that happens, everything is simply released. All right. So everything separates, the two subunits from the ribosome break apart, the transfer RNAs go bye-bye, and even the messenger RNA goes away. Okay. Now, this is obviously a video on YouTube, not me. So when you get to the end of this video, open up the PowerPoint and click on the hyperlink here and have a quick look at this. The illustrations and the animations are far better than my simple little videos here. Okay, now we're done talking about DNA and RNA. And there, but there's a couple of other things that I do want to mention here. All right, what we call today DNA technology or biotechnology. And so just a quick little look at some of the things that science is capable of doing with DNA these days. So over the course of the last couple of chapters, we've talked about things that can go wrong in a person's genome. All right, mutations in the genes or extra chromosomes or something. All right, and so we have learned over the course of the very recent past, in all honesty, to do what's called gene therapy. And what this is the insertion of genetic material into human cells in order to treat a disorder. And this can happen in two different ways. Ex vivo therapy, where you take cells out of a person, you alter them genetically, and then you return them to the patient. But there's also in vivo therapy, where the gene is directly inserted into an individual through a vector, like a virus. All right, we hear the word virus and we go, oh my God, that's terrible, right? We think COVID and death and destruction and everything. But in reality, some viruses are not terribly harmful. And so we can insert them into our bodies. And remember that viruses go right through the cell membrane and into the nucleus and start screwing around with your DNA. Remember, that's why some viruses are so dangerous. So what happens here is that the virus actually carries the gene that we want, the good gene, right into the nucleus of certain cells. And we do this to replace mutated genes or to restore a normal gene over the activity that is going on in a certain cell. Gene therapy has been very successful in treating some forms of cancer. Right, so you literally insert a virus with a gene that will help to control the cancer cells. Now, these are a couple of different terms that you hear thrown around on the news all the time, but a lot of people don't really know what they are. All right, and so let's define them. Genetic engineering. All right, this sounds like one of those science fiction things. Oh my God, we're going to create the master race, and we're going to, whatever. All right. In reality, all genetic engineering is the alteration of DNA in bacteria or viruses or plants or animals, right, through what we call recombinant DNA technology. And what this means is that we now have a DNA molecule, a gene, or even a chromosome that contains DNA from two or more different sources. All right, and I'll get into each of these in a little bit more detail a little bit later on. If you have inserted DNA into an organism, then we call that organism a transgenic organism. All right. And these are organisms that have a foreign gene inserted into them. Now, this brings up, before we get on to the last one on this list, uh, one of the things you hear, especially nowadays, especially in food sources, is the term GMO, genetically modified organisms. All right. And, advertisers, food salesmen especially, will try to tell you that their foods are not genetically modified. All right. And although they may not be genetically modified in a laboratory, almost all of our food items have been genetically modified. All right. The wild ancestors of cows did not give milk 365 days a year. Okay. They gave milk during the calving season. All right. Wild tomatoes are these teeny tiny little um, things that are about the size of a grape, and they taste like crap. All right. They're very bitter and awful. Um, so we have genetically modified tomatoes and corn and potatoes and all of the crops that we use. We've modified wheat. We've modified all sorts of things. So almost all of our food items are genetically modified, maybe not in a laboratory, but by the farmers or the ranchers. All right, you find a cow that gives milk more longer, I should say, than another cow. Well, you want that cow to breed. You can get rid of the other one. And so over time, we've changed the genetic makeup of our herds and our crops and everything else. All right, enough lecturing. All right, but my point is, you hear that term, genetically modified organisms, and which is what GMO stands for. And a lot of people don't understand that we have modified our foods. Last thing on this list is biotechnology, which is simply using natural biological systems to create a product or achieve a desired end. A good example of this is inserting a gene into a bacterial plasmid in order to get that bacterium to produce something that we want. A lot of the insulin that is produced synthetically in the United States these days for people with diabetes is actually produced by bacteria. We've inserted the human gene for insulin production into a bacterial plasmid, and the bacteria just produces insulin. It doesn't need insulin. It doesn't use insulin, but it produces it anyway. And so when we grow these in a big petri dish, and we can get the insulin out of the mix and then bottle it and sell it to your doctor's office or wherever you get insulin. A pharmacist, maybe? I don't really know. Um, I should know that, but I don't. Anyway, so that's an example of biotechnology. Okay, so gene cloning. All right, we've already mentioned that recombinant DNA is simply DNA that contains, or a chromosome or a gene that contains DNA from two or more different sources. And what it does is it allows genes to be copied. All right. And this is the example that I was talking about a moment ago, an example using bacteria to clone the human insulin gene. This is actually a fairly easy process. You find a restriction enzyme that's used to cut the vector or the bacterial plasmid and the human DNA that contains the insulin gene. Now, restriction enzymes are naturally found in most bacteria. They use them to destroy viruses. And what the enzymes do is they simply cut the DNA molecule into two or more pieces. We then use an enzyme called DNA ligase to seal together the insulin gene and the bacterial plasmid. The bacterial cell then takes up the plasmid. The gene is copied every time the bacterium reproduces, and a bacteria in a good environment can reproduce every 20 minutes. All right, and so every time the bacteria reproduces, you copy that gene, and then the bacteria are now producing huge quantities of insulin for no pay. All right, kind of a ripoff, actually. They should form a union. Okay, so how does that work? Well, we've got a human cell, and we find the chromosome that carries the human insulin gene, and that's shown in purple right here. Okay, we cut that out using a restriction enzyme in a process called cleaving, And we do the same thing for a bacterial plasmid. Now, remember, plasmids are not the DNA, heritable DNA from a bacterial strand. It's simply a ring of excess DNA, and the restriction enzyme cleaves it at the same spot. We then use an enzyme called DNA ligase to glue these two pieces together. And we formed a new piece of DNA, a new plasmid, which is now recombinant because not only does it contain the bacterial plasmid, but it also contains the human gene for insulin. We force the bacterium to take that recombinant DNA back into its cell. And then every time the cell reproduces, it produces another copy of the human insulin gene in a petri dish. And of course, those bacteria are now producing insulin. All right. We can then harvest the insulin and use it to help folks with diabetes. Okay. Um, Another thing that we can do, which is kind of interesting, is we can actually make copies of DNA molecules. And we do this by a process called PCR. Now, PCR stands for Polymerase Chain Reaction. And essentially, what it does is it's used to clone small pieces of DNA. And so you find a gene that you're interested in, or even a segment of a gene, and you cut it with a restriction enzyme and you put it in a little mixture. And then you go through a 3-step process, and I'm not going to go through all of the stages there. You have to mix in some new nucleotides and some enzymes and some other things. But essentially, it works like a DNA Xerox machine. It copies the DNA. So you start with the piece of the gene, and it copies the old strand with the new. All right. But then now the new is the old. And so in the third generation, the green represents the new. And in the fourth generation, the yellow represents the new. But notice that we are increasing the number of copies of this. And if you run this, 30 or 40 times, you can end up with billions of copies of a certain DNA molecule. The reason that this is important is because we need a large quantity of DNA to do certain kinds of analyses like DNA fingerprinting. All right, crime scenes and genetic analysis for a variety of things requires a large quantity of DNA. And that's impossible to get naturally because you need to harvest a great number of cells. In this way, all you need is a couple of cells, and you find this small piece of DNA inside those cells. And again, that's not a difficult process if you know the sequence of DNA and have an appropriate restriction enzyme. And so this process is used all the time. My students in general biology actually use this process along with the cloning process in the previous slide to insert a gene that makes bacteria glow in the dark. All right, the gene is found in jellyfish naturally, but we can insert it in bacteria and grow bacterial colonies that actually glow in the dark. It's a very cool lab. All right, so again, why do we use PCR? Well, primarily, one of the things that you probably are familiar with is DNA fingerprinting. All right, you see this on all the crime shows on TV all the time. And it's never as simple as they make it seem on TV, but it's not that difficult either. So what you do is you collect DNA from a crime scene, whether it's from the victim or possibly from a suspect, and you choose certain fragments of that DNA that are typically distinctive in different people. All right, in other words, they come in a variety of different alleles in people. And so let's say we have suspect A here. All right. And this guy, notice, has 12 repeats on this particular segment of DNA. Suspect B, has 12 repeats on one allele, but 16 repeats in the other allele. All right. Now, what we do is we insert these into a gel and then run an electric current through the gel. We stain the DNA so that it leaves bands, marks on the gel. All right. You run it for 45 minutes or an hour, and you get a pretty little pattern. So, um, if you have crime scene evidence with DNA that has two different alleles, you'll end up with two different bands in the crime scene DNA. If you've got two suspects and one of them only has one band at the 12 repeat mark, oh, come on, what's going on? There we go. Um, Sorry about that. I'm just going to keep going. So, 12 repeats um, at this mark, you only get one band. This suspect has one allele with 12 repeats and one allele with 16 repeats. He has two bands. That matches the crime scene evidence. And so we can use this in a court of law. DNA evidence has been acceptable these days. Anyway, that brings me to the end of the second of two videos on DNA and biotechnology, and I hope that you learned something.